In this next video, we're going to look at the stages of human development immediately following fertilization. Embryonic development begins with fertilization. This is the union of sperm and egg to form a zygote, and in a successful human pregnancy, this will occur typically within the oviduct. Copulation, or sexual intercourse, releases hundreds of millions of sperm into the vagina, but of those, only a few hundred survive the trip to the egg, and of those few hundred that make it to the egg, only one of them will end up fertilizing it. It was this statistic that led my high school anatomy physiology teacher to say, "Everyone's a winner, at least once in their life." If you think about those 500 million to 1 odds, you are the result of that sperm cell that made it to the egg cell. So if you're ever having a bad day, again, you can just think, everyone's a winner, at least once in their life. Now we can see in this electron micrograph image several human sperm cells colored in white. But what you may not realize is that the background, that spongy-looking material which the sperm cells are on, that is actually the surface of the egg cell. So you can see the egg cell is many, many times larger than individual sperm cells. A mature human sperm has a streamlined shape that suits its need to swim through fluids in the vagina, uterus, and oviduct. Now when we look at the structure of the sperm cell, there are 3 main parts. There's the head of the sperm cell, the midpiece, and the tail. So let's talk about each of these parts. The head of the sperm cell includes the nucleus, which contains the haploid chromosomes necessary for fertilizing the egg, but it also contains a vesicle full of digestive enzymes, known as the acrosome. So the acrosome, it's filled with these digestive enzymes that will help the sperm cell get through the zona pellucida, or jelly layer of the egg. We'll look at that in the next slide. The midpiece of the sperm cell contains many mitochondria. Now the reason why the sperm cell would need mitochondria is because it needs to power its swimming. It needs to be able to travel through the female reproductive system towards where the egg is located. And the tail will also assist with that swimming. It will provide the propulsion because the flagellum, which makes up the tail, is able to move back and forth in a whip-like manner. So here we see several points in time of a single sperm cell as it attempts to fertilize this egg. The sperm cell first has to make its way through follicle cells that might still be found around the outside of the egg. Once it gets past those follicle cells, it will come in contact with the zona pellucida, or jelly layer of the egg. This is where the enzymes within that acrosome become so important. If the sperm cell didn't have those, it would not be able to get through this layer. The first sperm cell to make it through the zona pellucida and come in contact with the plasma membrane of the egg, that is the sperm cell that will fertilize that egg. In fact, as soon as that first sperm cell comes in contact with the plasma membrane of the egg, it actually sends an electrical impulse around the surrounding of the egg, preventing any other sperm cells from fertilizing that egg. At this point, the sperm nucleus and the egg nucleus can fuse together to form a diploid zygote nucleus. This fertilization occurs within the oviduct, and the early stages of human development begin with fertilization and cleavage in the oviduct. So cleavage is a series of rapid cell divisions that occurs after fertilization. The key to development in all organisms is that each stage of development takes place in a highly organized fashion. Development after fertilization begins with cleavage. Cleavage is a series of rapid cell divisions that results in a multicellular ball. In humans, cleavage continues as the embryo moves down the oviduct towards the uterus. Typically 6 to 7 days after fertilization, the embryo has reached the uterus as a fluid-filled hollow ball of about 100 cells called a blastocyst. It is at this stage of development that the embryo will implant into the lining of the uterus. So again, about 1 week after fertilization. The embryo, which has become a blastocyst, implants itself into the uterine wall. One interesting aspect of the human embryo is that the outer layer of the blastocyst, the outer layer, is known as the trophoblast, and it becomes part of the placenta, the organ that will nurture and help the embryo develop while connecting with the mother's blood system, receiving nutrients and oxygen. Implantation marks the onset of pregnancy. Pregnancy, or gestation, is the carrying of developing young within the female's reproductive tract. In humans, it is measured as 40 weeks from the start of the last menstrual cycle. So it's not 40 weeks from implantation or 40 weeks from fertilization. It is 40 weeks from the start of the last menstrual cycle. Pregnancy is divided into 3 trimesters. Here we see a photo of a human embryo in the first trimester. By the end of the first trimester, the fetus looks like a miniature human being. The sex of the fetus can be determined by ultrasound, often near the beginning of the second trimester. Now, I asked my wife if it would be okay to share these pictures with the class, and she said it would. These are the first pictures of our son, Perrin. The first picture is at 9 weeks, and this was a confirmation of pregnancy appointment, and at the 18-week appointment, we discovered that we were having a son. The main developmental changes during the second and third trimesters involve an increase in size and general refinement of the human features of the fetus. So we can see already at 14 weeks, the general structure and shape, the human body plan, is definitely there, along with the development of fingers and toes, ears and eyes, nose and mouth. At 20 weeks, we can already see the formation of eyebrows and fingernails and toenails. At birth, a typical baby is about 50 centimeters, or 20 inches in length, and weighs 2.7 to 4.5 kilograms, or around 6 to 10 pounds. And here's a picture of our oldest son, Perrin, on the day he was born. Now, after birth, children develop quite rapidly. And so, our son continues to grow, and now his brother as well is continuing to grow and develop. The birth of a child is brought about by a series of strong rhythmic contractions of the uterus called labor. Hormones play a key role in inducing labor. These hormones are estrogen, oxytocin, and prostaglandins. This is one of the few cases of positive feedback that we see in human biology. Negative feedback is necessary for maintaining homeostasis, but positive feedback are usually responses which trigger themselves to occur more frequently. The contractions of the uterus are first started when estrogen from the ovaries induce oxytocin receptors on the uterus. Oxytocin comes both from the fetus and also the woman's pituitary gland, and this stimulates the uterus to contract. This in turn stimulates the placenta to make prostaglandins, which in turn stimulate more contractions of the uterus, which stimulate more production of oxytocin, which stimulate more contractions of the uterus. As you can see, this process would cause contractions to increase in frequency and intensity as labor progresses. The 3 stages of labor are dilation of the cervix, expulsion or delivery of the infant, and delivery of the placenta. The last topic that we'll discuss in this chapter is infertility. Infertility is clinically defined as the inability to have children after 1 year of trying. Now, just because a couple has not had a child in that first year it does not mean that they will never be able to have children. It just means that they are not as fertile, as typical, or as average. Infertility is most often due to problems in the man, such as underproduction of sperm or impotence. Female infertility is possible. But less common. Female infertility could result from either a lack of eggs in the ovaries or a failure to ovulate those eggs. Now, there are many technologies now available to help treat many of the different forms of infertility. Reproductive technologies can help solve problems related to the inability to conceive a child. One example of these technologies are reproductive pharmaceuticals. Everything from Viagra to medications which cause women to ovulate more than one egg each ovarian cycle. These pharmaceuticals can increase the likelihood of fertilization and implantation occurring. Probably the most extreme example of reproductive technology would be in vitro fertilization, in which eggs are surgically collected from the female combined with sperm collected from the male, and fertilization actually occurs outside of the body within a petri dish. Even though we have the technology to allow fertilization to happen externally, these fertilized eggs would then need to be implanted back into the uterus of a mother for the duration of pregnancy. So with that, we've come to the end of our discussion of human reproductive biology. In our next videos, we'll talk about DNA as the genetic material, how it was discovered, and how it functions. See you in the next videos.